The Assistant Professor, Department of Microbiology, Sri Lakshmana Institute of Medical Science, Pondicherry. So I'm going to discuss about antimicrobial resistance. So before going into the antimicrobial resistance, we'll see what are antimicrobial agents. Antimicrobial agents are the agents that kill or inhibit the growth of microorganisms. So it's nothing but an agent with kill or inhibit the growth of microorganisms known as antimicrobial agents. They're classified into various ways according to the microorganism against which they are used. From which they are using. If we are using against bacterial, it is antibacterial. If we are using against fungal organism, it is antifungal. If we are using against a parasite, it is an antiparasite. If we are using on any virus, it is antiviral agent. So depending upon the what we are using, whether we are using on bacterial, fungal, parasite, and viral. According to the ability to kill or inhibit the microorganism, it's classified as bactericidal, which is ability to kill. So this will kill the microorganism. Bacteriostatic means it will inhibit the growth of microorganism according to the source. So antibiotics are the natural substances which is produced by certain group of microorganism. Okay, so it is not. It's one of the natural substance produced by only certain group of microorganism. Whereas the chemotherapeutic agents, these are the agents which are chemically synthesized. So according to the action and usage, disinfectant is the one which destroys the wide range of the microbes present on the non-living surfaces to prevent the spread of infection. So disinfectant is nothing but it destroys the wide range of the microbes on the non-living surface to prevent the spread of infection. Antiseptics, which is mainly applied to the living tissues and help to reduce the infection during the surgery, like COVID and iodine. So it can be applied before the surgery to drape the abdomen. Okay. Then antibiotics destroys the microorganism which is present inside the body. So orally we have to give this like a form of tablet. So this will kill the microorganism which is present inside the body. Either it is available in a tablet form and IV, that is intravenous injection. So this is according to the action and usage. According to the chemical structure and mechanism of action, here the antimicrobials are further classified into many classes. So this is the mechanism of action, and we'll see what are the antimicrobial agents first. So this is divided into groups. The first one is a cell wall synthesis inhibitor. The second one is a cell membrane inhibitors. Third one is a metabolism inhibitors. Fourth one is DNA gyrase inhibitors, and the fifth one is protein synthesis inhibitor. So first, under cell wall synthesis inhibitors, beta-lactam antibiotics will come. So under beta-lactam, there's a certain group of antibiotics will be there. So one is penicillin, cephalosporin, and third one is carbapenem, and the fourth one is monobactam. Then glycopeptides, phosphomycin, cycloserine, and bezitracin. Then second one is cell membrane inhibitors. So under cell membrane inhibitors, daptomycin, polymyxin B, and colistin. So these are the group of drugs will come under cell membrane inhibitors. So this will inhibit the cell membrane. Okay. Then metabolism inhibitor. So this will inhibit the bacterial metabolism. So under this sulfonamides is one of the drug. Then coming to fourth one, that is a protein synthesis inhibitors. So under this tetracycline, chloramphenicol, macrolides, clindamycin, aminoglycoside, and quinapistin. So these are the group of drugs which inhibit the protein synthesis occurring in the bacteria. Then one more is a DNA gyrase inhibitors. So under this fluoroquinones and nalgesic acid. So this will inhibit the enzyme DNA gyrase. So coming to bactericide. So depending upon the killing, aminoglycosides and fluoroquinones. Time dependent. So this is concentration dependent killing. This is time dependent killing. This classified into two types. One is concentration dependent killing. Another one is a time dependent killing. So under concentration dependent killing, aminoglycosides and fluoroquinones, these are the two drugs will come under this. Under time dependent killing, beta-lactams and vancomycin will come under time dependent killing. So coming to antimicrobial resistance. So it is a development of resistance to an antimicrobial agents by microorganism. So it can be of two types, one is acquired and intrinsic. So it is a development of resistance to the antimicrobial agents which is produced by microorganism. So acquired resistance, it is classified into two types, one is an acquired and another one is an intrinsic. So this is a acquired resistance, what we are going to see now. So this refers to emergence of resistance in bacteria which is ordinarily susceptible to one group of antimicrobial agents by acquiring the gene coding for resistance. So this bacteria will be previously will be susceptible to one particular group of drug. Now it turns into resistance by acquiring a gene coding for resistance. So most of the antimicrobial agents shown by this are showing this type of resistance. So most of the group of bacteria showing this type of resistance, that is an acquired resistance. So they acquire the resistant genes and making the particular antibiotic drug resistance. Coming to emergence of resistance, one of the major problem worldwide in antimicrobial therapy. So infections by the resistant microorganism fail to respond to the standard treatment. Okay, then resulting in prolonged illness, higher healthcare expenditure, and greater risk of death. So infection, mainly the infection will be causing by resistant microorganism. So they won't respond to the normal standard treatment. Okay, resulting in making prolonged illness will be getting prolonged, and the higher healthcare expenditures and leading to greater risk of death. So mainly this is due to overuse and misuse of antimicrobial agents. It's one of the important factors for the development of resistance. So the evolution of the resistance strain, it's a natural phenomenon which occurs among the bacteria, especially when the antibiotic is overused. When we overuse the bacteria, this resistance strain will be formed naturally. Okay, so this makes bacteria won't respond to the particular drug when we are using the antibiotic overuse. Okay. Then use of particular antibiotic. So suppose we are using a particular antibiotic over a long period of time to the particular group of bacteria, this will promote the resistant bacteria to thrive and susceptible bacteria to die off. Okay. So one is the overuse and misuse of antimicrobial agents, one of the important causes. It's a short summary now for the development of acquired resistance. This evolution of the resistance strain is one of the natural phenomena because this occurs among the bacteria when the antibiotic is when we're using the antibiotic more for a longer period and using a particular antibiotic for a group of particular bacteria, which turns promotes the resistant bacteria to thrive and the susceptible bacteria to die off. Coming to intrinsic resistance, it refers to innate ability of the bacterium to resist a class of antimicrobial agents due to structural and functional characteristics. Here, it's a bacteria having some innate ability. So it has some ability to resist a particular group of antimicrobial agents due to its functional and structural characteristics. For example, in case of gram-negative bacteria, it will be resistant to vancomycin. So we won't give vancomycin in gram-negative bacterial infection. So this imposes a negligible threat as it is a defined pattern of and it's a non-transferable. Here the resistance cannot be transferred from one bacteria to another. However, the clinicians must be aware to exclude this antibiotic from the therapy. So they should be excluded while prescribing any antibiotic to the, if the patient is having some gram-negative infection, should not prescribe vancomycin to the patient. Okay. Coming to mutational resistance, resistance can develop due to mutation of the resistant gene. Okay, so here the resistance is due to mutation in the resistant gene itself. Okay, so this is typically seen in Mycobacterium tuberculosis developing resistant to anti-tuberculosis drug. So here, that's why we are using a group of combination of the drug in case of Mycobacterium tuberculosis. We are not using a single dose therapy. Transferable drug resistance. In contrast, transferable drug resistance is plasmid coded, usually transferred by conjugation or rarely by transduction and transformation. So it is here the resistant genes will be transferred from one bacteria to another bacteria, either by conjugation, transduction, or by transformation. So now we'll see one by one in detail. So these are the types of horizontal gene transfer. The first one is the transformation. Okay. So in transformation, that is the resistant gene which is present in the environment, that is naked DNA. So this has been transferred to the bacteria. So this bacteria will acquire this naked gene, that is for the resistant gene, and become resistant to particular antibiotic. So this is known as transformation. So this will be incorporated into the bacterial chromosome and makes that particular bacteria resistant to the drug. Coming to conjugation, here the resistant gene will be transferred from one bacteria to another bacteria by conjugation tube. Coming to the third one, that is the transduction. Here the resistant gene will be transferred from one bacteria to another bacteria by a vehicle known as bacteriophage. Bacteriophage is nothing but virus infected bacteria. So from this bacteria will be getting resistant. That is one, the resistant gene will be carried to another bacteria and making a particular bacteria resistant by a through vehicle known as bacteriophage. Coming to transformation, as already we discussed, it's a process of random uptake of free or naked DNA fragment from the surrounding. So the randomly the bacteria will take the free or naked DNA fragment from the surroundings and make them incorporated into its DNA fragment. From there, it enters into the chromosome and make it resistant. This is known as transformation. Coming to transduction, it is a transmission of portion. Only the portion of the DNA is being transferred from one bacteria to another bacteria by a vehicle known as bacteriophage. The bacteriophage is nothing but virus-infected bacteria and multiplies inside the bacteria. Coming to conjugation. Conjugation is the transfer of genetic material from one bacteria to another bacteria by meeting or contact with each other by forming a conjugation tube. So coming to mechanism of antimicrobial resistance. Now we'll see what are the mechanisms which is involved in antimicrobial resistance. So these are the four important mechanisms which has been involved in mechanism of antimicrobial resistance. First one is the decreased permeability across the cell wall. So this is the decreased permeability across the cell wall. Second one is an efflux pump. Third one is an enzymatic inactivation. And fourth one is by modifying the target site. So in which this is a picture showing the antimicrobial resistance. So decreased permeability across the cell membrane. Okay, first one is the decreased permeability by across the cell membrane. Second one is the efflux pump. Third one is by enzymatic inactivation, then amplifying the target site. So the target site is getting amplified. And so the first one is showing the protecting the target site. Either the target site has been getting protected or the amplifying the target. So the target will be getting amplified. So it won't fix, the antibiotic won't fix to the particular target site. Then preventing the antibiotic entry. Once the antibacterial enters into the cell membrane, it will prevents the entry of antibiotics into the cell wall. Then efflux pump. That is the once the antibiotic, that is a drug enters into the cell wall. So this particular bacteria will efflux the drug. It will expel the drug outside. Then modify the antibiotic. And another thing is that it will modify the antibiotic. And third one, it will destroy a particular antibiotic. Or it will modify the target. So these are the important mechanism which has been involved in antibiotic resistance. Now we'll see one by one. So decreased permeability across the cell wall. Here certain bacteria will makes a change in the cell membrane poring channel, either in the form of size, shape, and preventing the entry of antimicrobial agents into the cell wall. So this resistant mechanism is observed mainly in gram-negative bacteria like Pseudomonas, Enterobacter, Klebsiella species against particular group of drugs like imipenem, aminoglycoside, and quinones. So first one is a decreased permeability across the cell wall. Here certain bacteria will modify the cell membrane poring channel will getting modified. So either in the form of size, shape, and preventing the antimicrobials from entering into the cell. So this resistance mechanism is seen in many gram-negative bacteria like Pseudomonas, Enterobacter, and Klebsiella. So this or the organism will be against the group drugs such as imipenem, aminoglycosides, and quinolones. Coming to efflux pump, that is the second one. Certain bacteria possesses efflux pump, which mediate the expulsion of the drug from the cell and prevents the entry of intracellular accumulation of the drug. So this strategy has been seen in Escherichia coli and other enterobacterials against tetracycline and chloramphenicol. So efflux pump is nothing but once a antibiotic enters into the cell wall, bacterial cell wall, it will immediately expel the drug from the cell wall and preventing the entry of intracellular accumulation of the particular drug, antibiotic drug. So this strategy has been seen in Escherichia coli and enterobacterials against a particular group of drugs, antibiotic drugs like tetracycline and chloramphenicol. Staphylococci against macrolides and streptogramins, Staphylococcus aureus and Staphylococcus pneumoniae against fluoroquinolones. By enzymatic inactivation. Enzymatic inactivation, certain bacteria can inactivate the antimicrobial agents by producing various enzymes such as beta-lactamase enzyme production. So this is observed both in gram-positive and gram-negative bacteria. And so it will break down the beta-lactam ring, thereby inactivating the beta-lactam antibiotics. So aminoglycoside modifying enzymes like acyl transferase, adenyl transferase, phosphotransferase produced by both gram-positive and gram-negative bacteria. This will destroy the structure of aminoglycosides. Then modifying the target site. Modification is the in the target site of antimicrobial agents, a very important mechanism. So the particular target site for the antibiotic to getting modified. This is especially seen in MRSA, that is methicillin sensitive Staphylococcus aureus. In this strains, target site of the penicillin, that is a penicillin binding protein, getting altered to penicillin binding protein 2E. So the altered penicillin binding protein coded by the chromosomic coded gene MECK gene, they do not sufficiently bind to the beta-lactam antibiotics. Therefore, prevent the entry of prevent them from inhibiting the cell wall synthesis. So the beta-lactam resistance is seen in pneumococci is due to alteration of the penicillin binding protein to penicillin binding protein 2B. So this then streptomycin resistant Mycobacterium tuberculosis is due to modification of the ribosome, that is 16S rRNA. Rifampicin resistant in Mycobacterium tuberculosis due to mutation in the RNA chromosome, RNA polymerase. Then quinolone resistance seen in many gram-positive bacteria, like particularly Staphylococcus aureus and Streptococcus pneumoniae, mainly mutation in the DNA gyrase enzyme. Vancomycin resistance in enterococci. So this takes a change in the target size. So they will change the target site in the form of D-alanine side chain of peptidoglycan. So now we'll go for this small summary. What are the four important mechanisms which has been involved in antimicrobial resistance? The first one is the decreased permeability across the cell wall. So certain bacteria have a cell membrane poring channels, modify the cell membrane poring channel either in the form of size, shape, and preventing the entry of antimicrobial agents entering into the cell wall. This resistance is seen mainly observed in many gram-negative bacteria like Pseudomonas, Enterobacter, and Klebsiella species against a group of drugs like imipenem, aminoglycosides, and quinolones. Second one is an efflux pump. Certain bacteria possesses efflux pump which mediate the expulsion of the drug. Once the drug enters into the cell, it prevents the intracellular accumulation of the drug. So it will expel the drug outside the cell wall. So this is seen in Escherichia coli and Enterobacterials against tetracycline and chloramphenicol. And Staphylococcus against macrolides and streptogramins, Staphylococcus aureus and Streptococcus pneumoniae against fluoroquinolones. So this is about the efflux pump. Third one is by enzymatic inactivation. Certain bacteria inactivate the antimicrobial agents by producing various enzymes like beta-lactamase, then aminoglycoside modified enzymes. So beta-lactamase enzyme production, this is seen in both gram-positive and gram-negative bacteria. It will break down the beta-lactam ring by inactivating the beta-lactam antibiotics. Then aminoglycoside modifying enzymes is seen like acyl transferase, adenyl transferase, phosphotransferase. This is observed in both gram-positive and gram-negative organisms. It will destroy the structure of aminoglycoside. So the aminoglycoside group of drug won't act. Okay. Then by modifying the target site, the target site getting modified either this is observed in MRSA. So this is very important mechanism. And beta-lactam resistance which is seen in pneumococci. In MRSA, the target site has been getting modified to penicillin binding protein 2A. So it won't sufficiently bind to the beta-lactam antibiotics due to McKay preventing them from inhibiting the cell wall synthesis. Then beta-lactam resistance which is seen in pneumococci, it's due to penicillin binding protein getting altered to penicillin binding protein 2B. In MRSA, it has been altered to penicillin binding protein 2A, whereas in case of pneumococci, it has been altered to penicillin binding protein 2B. Okay. Coming to streptomycin resistance, which is seen in Mycobacterium tuberculosis, here there is a modification of ribosome in the form of 16S rRNA, and rifampicin resistance, which is seen in Mycobacterium tuberculosis due to mutation in the RNA polymerase, and quinolone resistance, which is seen in gram-positive bacteria, particularly Streptococcus and aureus and Streptococcus pneumoniae due to mutation in the DNA gyrase enzyme, and vancomycin resistance in enterococci in the form of the target site getting altered to D-alanine side chain of peptidoglycan. Coming to classification of beta-lactamase. The AMPER classification is one of the important classification which is seen in beta-lactamase. Now we see the class. Class A is an extended spectrum beta-lactamase, that is ESBL. The organism producing ESBL enzyme are resistant to all penicillins here. And first generation, second and third generation cephalosporin monobacter. The organism which is producing the ESBL enzyme, that is extended beta-lactamase, spectrum beta-lactamase are resistant to all penicillins and first, second generation, third generation cephalosporin monobacter, but remain sensitive to carbapenem and saccharomycin. So they be sensitive to only to carbapenem and saccharomycin. Class B metallobeta-lactamine, that is MBL. This organism are resistant to all antibiotics which are producing AMC. In addition, they are resistant to carbapenem. So here this is the organism resistant to all antibiotics which are producing the AMC. In addition, it is resistant to carbapenem. The resistance cannot be overcome by beta-lactam and beta-lactamase inhibitor combination. So this resistance cannot be overcome by beta-lactam and beta-lactamase inhibitor combination. So this will be detected by a test known as EDTA, a test synergy test and modified Hodge test. Coming to class C, there is an AMC beta-lactamase. Here the organism is resistant to all antibiotics to which ESBL producers are resistant, plus they are resistant to saccharomycin, that is cefoxitin and cefoprotidine, but sensitive to carbapenem. Resistance cannot be overcome by beta-lactam and plus beta-lactamase inhibitor combination. So in class C, the organism will be resistant to on antibiotics, which ESBL are producing, resistant plus they are resistant to sulfamycin. There is a sulfacetin and sulforaten, but sensitive to carbapenem. The resistance cannot be overcome by beta-lactams plus beta-lactamase inhibitor combination. Class D is the oxygenase. Oxygenase, that is the resistance can be overcome by beta-lactam plus beta-lactamase inhibitor combination. Coming to antimicrobial susceptibility testing. So this test will be done only for the pathogenic pattern, not for the commensal. So for example, if suppose if the E. coli, you are isolating the E. coli in the urine sample, antimicrobial susceptibility test will be done. And we will choose an antibiotic. Then for if suppose if the E. coli is been isolated from the stool sample, it is considered as commensal. Here we won't perform AST classification. So certain methods, many classic methods has been involved in antimicrobial susceptibility testing. So these are the tests which has been involved. First one is a distribution method. And the distribution method, Kirby bar distribution method, Stokes distribution method is there. Then second one is a dilution test. That is a broad dilution method, agar dilution method are there. Then third one is an A test, and fourth one is an automated method. The last one is a molecular method, PCR detecting the drug resistant gene. Now we'll see one by one. So under Kirby bar distribution method, so this is a diagram showing how they are doing a distribution method. So first they are taking the sterile nutrient agar, then they are pouring the organism to be cultured. Then antibiotic disk has been kept 6 mm apart. So only 6 drugs has been supposed to kept in one particular antibiotic that is in inter agar. Then we have to incubate that plate in 18 to 24 hours for 37 degrees centigrade. Then antibiotic will diffuse in agar radially. The zone of inhibition is taken as an organism is resistant to. If there is no zone of inhibition around the particular antibiotic dose, it is taken as a resistant. If there is a zone of inhibition, that is indicates organism is sensitive to particular antibiotic. So this will be measured by Vernier scale. According to that only, we have to choose which antibiotic is sensitive and which is resistant. So distribution method is a commonly used method, mainly suitable for rapidly growing pathogenic organ bacteria. It's unsuitable for slowly growing organism. Here the filter paper disc, which is implicated with appropriate concentration of antibiotic solution, is being used. So the test bacterium has been inoculated into the solid medium and antibiotic disc was kept 6 mm apart. The antibiotic disc diffuses into the solid medium. So the concentration is the highest near the site of application of the antibiotic disc and the decreases gradually away from it. Here the sensitivity of the drug is determined by zone of inhibition of the bacterial growth around the disc, particular antibiotic disc. Then we choose a Muller-Hinton agar. So it's one of the best medium for routinely used for susceptibility testing for fastidious bacteria, mainly for the two reasons. It shows batch-to-batch susceptibility, reproducibility of the susceptibility testing and supports the satisfactory growth of non-fastidious pathogen. So for this purpose only, we're choosing the Muller-Hinton agar. Coming to inoculum. Inoculum, there's these are the protocols should be followed. So here the isolated pure colonies of the test organism has been inoculated into the suitable liquid medium, either in a peptone water. Then it should be incubated at the, and the turbidity should be adjusted with the Parkland capacity. That is, capacity should be turbidity will be adjusted with according to 0.5 Parkland. Okay. Density should be equal comparing. Then the lawn culture has been made in the overnight incubator broth and inoculated into the Muller-Hinton agar medium by separating with the spreading with the sterile cotton swab. Then if the inoculum should not be too heavy, okay, and too light, should be medium, and it shows the inhibition of inhibition zone properly. Then these are the control stains has been used. Similar to the test inoculum, control stains has been made. Test for AST. So these are the control stains has been used. Escherichia coli ATCC 25922. Then Pseudomonas aeruginosa ATCC 27853. Streptococcus aureus ATCC 25923. Enterococcus faecalis ATCC 29212. Coming to antibiotic disc. So it should be commercially available and prepared in-house. Then sterile filter paper disc of 1 mm has been integrated with the standard quality solution of antibiotic. Then antibiotic disc should be chosen, it should be commonly used for the therapy. Then particular organism what we are testing, it should be used. Then resistant pattern of the locally prevalent pathogen should be known. So by this only we have to choose an antibiotic disc. So one is by it should be used for the therapy and the organism should be against which drug should be tested. Then the third one is the resistant pattern of the locally prevalent pathogen. Coming to second one, there is a Kirby bar distribution method. So here the cotton swab is dipped into the inoculum and squeezed to drain out the excess fluid. Then the swab has been inoculated into the Muller Hinton agar plate for sticking it 3 times over the entire agar. Once the agar plate is getting dried, the antibiotic disc has been kept by a sterile forceps or with multi-disc dispenser. The disc should not be kept closer. Up to 6 discs has been kept in the 100 plate, and then the plate has been incubated for 37 degrees centigrade for 16 to 18 hours. So this is a picture showing how we'll do a Kirby bar distribution method. Zone of inhibition is measured by using a ruler or vernier caliper. The control sign should be tested during each time when a new batch of disc or Muller Hinton agar is used. Okay, so this is a bacterial suspension, and then they're spreading this over the Muller Hinton agar by a sterile spap stick 3 times. Then we are keeping the antibiotic disc 6 apart by a sterile forceps at this dispenser. Then they are incubating at 37 degrees centigrade for 24 to 48 hours. Then next day, they're seeing the zone of inhibition by using a vernier caliper or ruler. So this is a picture showing the antibiotics. Around this zone of inhibition indicates bacterial growth. So this is considers a resistant. There is no zone of inhibition with a particular antibiotic disc. And this drug is showing some zone of inhibition against the antibiotics. So this drug is being susceptible, and it's showing some zone of inhibition in a minimal, that is a moderate amount, it taken as an intermediate. So by this, they will give an antibiotic. So this is one more picture showing how they are doing the Kirby bar distribution method. Here the MHA plates has been divided into, this is coming to Stokes distribution method. In here, the MHA plate has been divided into 3 parts. The test organism has been inoculated into the center 1/3, and the control organism in the upper 1/3, and the lower 1/3 of the plate. The test organism will be inoculated in the center 1/3. Control will be inoculated in the upper and lower 1/3. Okay, reporting. How the reporting will be done? The sensitivity report is prepared by comparing the zone of inhibition. The zone of inhibition in the control and test bacterium will be compared here. Sensitive indicate zone radius is wider than or equal not than 3 smaller and control. Intermediate zone radius is 2 but smaller than control by more than 3 Resistant, no zone of inhibition. Coming to primary distribution test. So in Stokes distribution method, the MHA plate has been divided into 3 parts. So the first test inoculum has been inoculated in the center 1/3, the control one in the upper and lower 1/3 of the plate. And reporting should be done comparing the inhibition zone in zone control and test bacterium. Sensitive indicates zone radius is wider or than equal, no more than 3 smaller than control. And intermediate, the zone radius is 2 but smaller than the control by 3, more than 3 Resistant, no zone of inhibition. Okay, so this is how they will report in Stokes distribution method. Coming to primary distribution test, this is performed when the results are required urgently and single pathogenic organism is being in a particular sample. Here the clinical sample has been directly inoculated uniformly into the surface of agar plate, and antibiotic dose has been kept applied. Coming to dilution test, here the antimicrobial agents is serially diluted in each dilution. The test organism has been inoculated, and antimicrobial susceptibility test has been done, and MIC is calculated. The MIC is nothing but a lowest minimum inhibiting concentration of the drug. So it is a lowest concentration of the antimicrobial agents that inhibit the visible growth of the microorganism after an overnight incubation. So this is how they will perform a MIC. So test organism is inoculated in the tube containing a serial dilution of antibiotic. So this is the control one. So here they're serially diluting the antibiotic solution, serially diluting the antibiotic, and the fixed amount of the test organism has been inoculated into all the tubes. Next day, we will see that this tube has been incubated for 24 to 48 hours for 37 degrees centigrade. The next day, we will see the turbidity. The turbidity should be compared with the control. Okay, then MAC is calculated as a minimum inhibitory concentration. Should not be any growth in there is a lowest concentration of the antimicrobial agents will inhibit the visible growth of the microorganism is taken as an MAC. Then agar dilution method. Here the serial dilution of the agar drug has been prepared in a molten agar and poured into a petri dish. The stain has been test stain has been spot inoculated. This method is more convenient than broth dilution method. Here advantages, several stains will be tested at the same time by using a stain plate. Coming to antibiotic assay in body fluids. So mainly done to assess the toxic level and therapeutic level of the drug in the blood and other body fluids. That is known as antibiotic assay in body fluids. Both the dilution and distribution method can be tested against a standard suspension of the bacteria with the known MIC or zone of inhibition. So this mainly done to assess the toxic level and therapeutic level of the drug which is present in the blood and other body fluids. So both dilution diffusion method can be followed by testing against the standard suspension of the bacteria with the known MIC and zone of inhibition. Coming to ellipsometer or E-test, this is a quantitative method of detecting MIC by using a principle of both dilution and diffusion of antibiotic into the medium. So it uses the absorbance strip, which contains a predefined gradient of antibiotic concentration, which is immobilized along its length. So it has been applied in the long culture of the bacterium. Okay, following incubation of the test organism, elliptical zone of inhibition has been produced around the particular antibiotic strip. The antibiotic concentration at which the eclipse edge intersects the strip is taken as an MIC. So ellipsometer is nothing but it's a quantitative method for detecting the MIC by using the principle of both dilution and diffusion of antibiotic into the medium. It uses an absorbance strip which contains a predefined gradient of antibiotic concentration which has been immobilized in the along its length. Okay, so it has been applied as a lawn culture lawn inoculum of the bacteria. So this strip has been inoculated into the lawn inoculum of the bacteria. So following incubation of the test organism. So the next day, if this should be incubated at 37 degrees centigrade for 24 to 48 hours. Next day, we will see a incubation of the test organism. That is the zone of elliptical zone will be produced surrounding the antibiotic strip. So antibiotic concentration at which the eclipse edge intersects the strip is taken as an MAC value. So this is the picture showing the how they are doing the ellipsometer. That is the E-test. So this is the lawn culture which is produced by the test bacterium. They are making the lawn culture, the Muller and Hinton agar, right? Then the antibiotic strip has been applied in the center by using a sterile forceps. The next day they are seeing the zone of inhibition, that is an elliptical zone which intersects. So this they taken as an MAC. Here, the one of the advantages, the dilution and diffusion method of seeing the same test. So this is one of the advantage in this test. Coming to automated antimicrobial susceptibility testing, several automated methods are available as a VITAC2 bacterial identification and antimicrobial sensitivity system, Phoenix system, microscan walkaway system, MOST system or computer assisted, and have a sophisticated software to analyze the growth rates and determine the antibiotic susceptibility report. They use commercially available panels which contain added growth factors to speed up the growth of the organism. Coming to that is the last one, that is a molecular method. The PCR-based assays are available targeting the specific drug resistant, for example, MECI gene for MRSA detection. So coming to antimicrobial stewardship program. So introduction, the optimal selection and dosage duration of antimicrobial agents results in best clinical outcomes. So before choosing antibiotic, we have to choose a correct drug to the patient to the correct time at the correct dose for the correct duration for preventing the infection with minimal toxicity to the patient and minimal impact on subsequent resistance to prevent the resistance. This is how we have to choose an antibiotic. So the program is mainly based on how to choose an antibiotic to the patient. Okay, definition according to CDC, that is prescribing the right antibiotic for the right patient for the right time with the right dose with the right route causing least harm to the patient is known as. So this is the main aim of this antimicrobial stewardship program. Then coming to de-escalation. What is meant by de-escalation? Reduction in the spectrum of antibiotic, administered antibiotic to the discontinuation of antibiotic or switching the agents from narrow spectrum once the pathogen is identified and sensitivity of known. Suppose if we are using antibiotic, suppose if the patient has been sick in the ICU and we are giving an empirical antibiotic treatment, once a report, the culture report arrives, we have to choose that is de-escalate the drug from broad spectrum to narrow spectrum. So day one, we are giving the empiric treatment, that is empirical antibiotic treatment without knowing which factor has been involved. Then the day three, it has been changed to de-escalation, that we have to broad spectrum to narrow spectrum. Have to choose antibiotic. Then antibiotic timeout is nothing but review by reassess the empiric antibiotic after 48 to 72 hours of the initiation. Then coming to antimicrobial policy, prepared using a following, this has been prepared by using a general principles like Data is analyzed on quarterly basis, then standard treatment guidelines, prescription auditing, review of surveillance data generated from antibiograms and prescription auditing, education and training of all infection control activities in collaboration with the HACC. So this is about the antimicrobial policy. Here the data should be analyzed on quarterly basis. Standard treatment guidelines should be followed. Then a prescription auditing should be done. Review of surveillance data. You have to review the surveillance data generating the antibiogram and prescription auditing. Then education and training for all the infection control activities in collaboration with HACC. So HACC is nothing but hospital infection control. Here the chairperson, that is the head of institution, that's a medical superintendent, is a chairperson. Secretary is a clinical microbiologist, infection control officer, infection control nurse, epidemiologist, equivalent medical faculties, hospital kitchen in charge, clinical pharmacist, nursing superintendent, operating room manager, the director of environmental services. Coming to hospital infection control policies, so certain guidelines for the prevention and control of infection, antimicrobial policy, surveillance policy, disinfection policy, and isolation policy for investigation of an outbreak of infection. HACC means should be conducted regularly, once in a month or often when it is being required. When there is an outbreak and if any infection should be conducted. So coming to infection control, healthcare associated infection under this UTI, pneumonia, SSI, and BSI. Bloodstream infection, CAR-T, that is catheter-associated urinary tract infection, ventilator-associated pneumonia, then CRBSI, that is catheter-related bloodstream infection, and surgical site infection. This has been considered under healthcare-associated infection. Hand washing is one of the key component in preventing the infection control. Okay. Coming to hand wash, how we perform a hand wash or hand rub. So first one, we have to get a hand rub from this palm to palm. Then third one is the back of palm, both sides, interlaces, that is in between the web spaces. Then fifth step is the back of fingers on the palms on both sides. Then sixth step is a thumb, rotating the thumb, rubbing the thumb. Then seventh step is the scrubbing of nail beds on the palms on both sides. So these are the 5 moments of hand hygiene. So there is a difference between moments of hand hygiene and steps in hand wash. So these are the 5 moments of hand hygiene. So first one is before touching the patient, after touching the patient, then before doing any procedure, and after doing the procedure, and after touching the patient surrounding. So these are the 5 moments of hand hygiene. So coming to the conclusion, endemic MDR is effectively controlled by strict antimicrobial stewardship program and comprehensive infection control measures. Thank you.